Make the ground shake.
African sleeping sickness is a vector-borne parasitic disease caused by the parasite Trypanosoma brucei, or T. brucei. The infection is spread by flies which have acquired their infection from humans or animals harbouring the parasites. In the first stage of infection, the parasites multiply in sebaceous tissues, blood and lymph of its host. This causes minor symptoms such as fever, headaches, joint pains and itching. In the second and more serious stage, the parasites cross the blood-brain barrier to infect the central nervous system. This is when the more obvious symptoms of the disease appear, such as changes in behaviour, confusion, sensory disturbances and poor coordination. Disturbance of the sleeping cycle, which gives name to the disease, is an important feature as well. Without treatment, sleeping sickness is considered fatal. This study was conducted to address a severe lack of treatments for this life-threatening disease. The research group focused on inhibition of T. brucei N-myristoyltransferase, or TBNMT, as it is a promising therapeutic target for this disease in infected mice. Protein N-myristylation is an essential modification which is necessary for T. brucei survival, thus making it a good drug target.
So one of the key elements of sleeping sickness is the parasites you kind of get bitten by say the tsetse fly, and to start with it just circulates in your bloodstream. But after a while, and it depends on the actual strain of sleeping sickness how long this takes, but then it gets into the brain, and that's when it starts to affect your brain, and that's when you get the neurological changes that gives it its name. So your sleeping circadian rhythms get disrupted, and you don't sleep in the night, you sleep in the day and things like that. And the other problems and what eventually kills you. And what makes it difficult is you've got to get the drug into the brain, and that's not always easy. You know, obviously your brain is a very sensitive part of your body, and you want to keep toxins out of your brain. So you have something called the blood-brain barrier, which stops things getting in. You also have other systems that pump compounds out that get in. And so what you have to do is have compounds that can kill the parasite, have a mechanism to kill the parasite, and you have to get it into the brain, and you have to stop it being pumped out. So get all of that together, but also stop having other off-target toxicities. It's a very important enzyme across a lot of organisms, because what it does is puts a C14 fatty acid onto the end of a lot of proteins, and so that has quite a few functions. It activates some proteins, it can stabilize proteins, but also if you imagine you've got a fatty greasy bit on the protein, and that helps to anchor it into membranes, which are also fatty acid based, and so it can change the localization of proteins from, say, the bulk of the cell into the nucleus, and therefore the action of that protein, if it's an enzyme, it can localize that protein, it can activate it, it can stabilize it.